My name is Ben Sonnenberg. I'm a PhD candidate at the University of Nevada, Reno, and I'll be talking about developmental stress and spatial cognition in a wild food-caching bird. Understanding how heterogeneous environments shape organisms during their development, resulting in differing phenotypes, and how this phenotypic variation manifests across populations and environments is of critical importance in ecology. How development shapes complex phenotypes, such as cognitive phenotypes, has mainly been studied in the laboratory environment, where it is focused on model or near-model organisms, and all of these studies have a main pattern, which show that experimentally induced stress results in future cognitive impairment. But do these patterns hold in wild systems? Mountain chickadees reside in western North America in montane environments. We study a population in the Sierra Nevada mountains at the Seychan Experimental Forest in California. Chickadees are scatter-hoarding species that cache tens of thousands of food items in order to survive the harsh winter months of the Sierra Nevada. We study a population of chickadees that stretches across a montane elevation gradient that is characterized by differences in environmental harshness. Two of our study locations are situated across a snow line. A snow line is an approximate elevation in our field site, 2,100 meters, that during transitional periods, such as spring and fall months, precipitation is more likely to fall above the snow line as snow and below the line as rain. This translates into higher elevations experiencing more snow and colder temperatures for a longer period, and thus being considered an overall harsher environment comparing to low elevations, which allows a unique comparison between individuals residing at each location. Our low elevation location is at approximately 1,900 meters, and our high elevation site is at 2,400 meters. Caching has long been hypothesized to be critical for overwinter survival, especially in harsher environments. And in past comparisons between elevations, we've shown differences in hippocampal morphology, with higher elevations having larger hippocampal neurons, which correspond to more caching at higher elevations, as well as better performance on a spatial learning task. Additionally, in recent years, we've shown that juvenile birds with better spatial memory performance are more likely to survive the winter months, which is direct evidence that this trait is under selection. We are able to test learning and memory in wild chickadees by individually tagging birds with passive integrative transponders, or PIT tags, the large white and purple tags of the two individuals on this slide. This allows them to interact with radio frequency identification, or RFID-equipped feeders. For example, when individuals land on the black antennas on the outside of a feeder mechanism, if they land on a correct feeder, a door will open for them, allowing them access to a food reward. There are 4 arrays across our site, 2 at each elevation, that are comprised of 8 individual feeders arranged in a classic array format. Each time chickadee visits an array is considered a trial, and the trial ends when the bird has located its rewarding feeder. If a chickadee visits an incorrect feeder, this is considered an error, and errors are averaged across the trials of the testing period before use in analyses. Levels of corticosterone were used to assess differences in development between individual chickadees. Blood plasma samples are a commonly used method for detecting circulating cort. However, such samples really only give a single data point for the entire developmental period, so we chose to use feathers. Chickadees grow their tail feathers in particular across the entire early developmental period. The majority of the feather is grown while the birds are still in the nest. These feathers provide a cumulative summary of cort levels across development. Mature tail feathers from first-year juvenile birds, as they were abandoned in the fall months, were used in a methanol-based extraction, followed by a standard ELISA assay to assess the total amount of cort contained in each feather. We predicted that there would be a large variation in levels of corticosterone between and within elevations, but with higher elevations showing lower levels of corticosterone. We also predicted that higher levels of corticosterone would correlate to poor performance on a spatial learning and memory task within elevations. We did see a large variation in corticosterone deposition across years. On the y-axis is corticosterone picograms per millimeter of feather. And the purple circles show high elevation and the green triangles show low elevation. There was no differences in elevation across all 3 years of the study. However, there was a significant difference in 2020 with 2020 showing much lower corticosterone deposition on average than 2018 and 2019. We also did find a relationship between court deposition in the tail feathers and performance on a spatial learning and memory task, but only within a single year. And this was only actually at a single elevation, as we only had 2 samples for the low elevation site for 2018. But we did find a very strong relationship, and the relationship as we predicted it, with birds with more court deposition in their tail feathers showing poorer performance on a spatial learning and memory task. However, the strong relationship disappeared in the 2 following years of 2019 and 2020. On a second related cognitive test, reversal learning, which tests flexibility, we saw no relationship in either 2018, 2019, or 2020 on performance on this task and deposition of court in the feathers. In summary, court showed a large variation between years, with 2020 having the lowest levels. However, there are no differences in court deposition between elevations across the years of the study. We did find that higher levels of court correlated with worse performance on a memory task, but only in a single year in 2018, and then the pattern disappeared. So how do we explain these patterns of variation? Well, it is very likely due to local climate, namely snow levels, as these are very tied to the year-to-year food availability, not only the abundance and diversity of available food, but the timing of when that food appears. And to give you an example of what yearly climate looks like in regards to snow levels at our field site, this is 2017 through 2020 with snow depth included on the y-axis. And I include 2017 as it is the highest snow levels that our field site has received in over 100 years. And it has been hypothesized that there was a large negative carryover effect for many insect taxa in 2017 that would have carried over into the spring of 2018 when chickadees would have been feeding their offspring, and the only year in which we saw an effect on chickadees and a relationship between cognition and levels of corticosterone. So we believe that one possible answer is that there are years that lack certain prey items, and these prey could be critical for dealing with stressors during development. For example, spiders contain a large amount of a substance called taurine, which has been shown to act as a neuroprotectant during development in laboratory studies. And a close relative of chickadees in Europe have been shown to preferentially seek out spiders to feed their young. Overall, the association between developmental stress and spatial cognition is not consistent across conditions, but that could be related to the fact that food caching species may have evolved a mechanism involved in the protection of spatial cognition that could be related to key components of their diet. With that, I'd like to thank you guys for listening. Thanks for my co-authors and all my funders.